Assalamu alaikum and welcome. In this tutorial, we are going to discuss about bioenergetics, the second topic of PM and DC. First of all, we have to know about bioenergetics, which is the quantitative study of relationship of and energy relationship in a biological systems, and it obeys the laws of thermodynamics. Then came forward. Our life organism need energy, and these energy come from carbohydrates and proteins, fats, lipids, and basic nutrients needed by organisms and by the process of photosynthesis and respirations. By photosynthesis, new products are formed, and by catabolisms and respirations, new products are broken down to form ATP. ATP is a kind of chemical link between catabolism and anabolism. It is important landmarks and important points. So carbohydrates, photosynthesis and respiration happen. Understanding bioenergetics, biological systems. Photocarbohydrates, in which energy is stored, energy is preserved, and glycolysis and respiration is the process whereby the energy is stored is released in controlled manner. So carbohydrates are energy storing, and respiration and glycolysis are energy releasing processes. Then came forward photosynthesis. Photosynthesis is the process by which carbon dioxide and water combine to form glucose, oxygen, and water. Water is act as a reactant in both reactant product and appear at both sides. It is with the help of chlorophylls. If we balance these chemical equations, we came across the chemical equations, that is this 6CO2 6H2O plus light energy is equal to C6H12O 6 plus O2. Carbon dioxide form glucose. It is the exactly opposite of the respirations. If glucose and oxygen are used to form the carbon dioxide and water, this is called respirations. Here is the respiration and here is photosynthesis. Photosynthesis uses the product of respiration, and respiration uses the product of photosynthesis, and where respiration occurs throughout day and light, and respirations occur only at daytimes. Compensation point is the important point at which the rate of photosynthesis and respirations become equal for some times, and it occurs mostly at dawn and dusk when the light intensity is below normal. When light intensity become greater, the photosynthesis become high, and carbon dioxide become low, and oxygen become high. Oxygen is sent to the atmosphere, and carbon dioxide is absorbed from the atmosphere. And this oxygen that is erupted in photosynthesis came from water, not from carbon dioxide, and it was given by Van Neal and was proved when the oxygen of radioactive isotope O18 was used. In first group of plant, they use the water containing O18s that produce oxygen O18s, but not the second group, in which CO2 containing O18 was given, but it produced only O2, not with O18. So the source of oxygen came from photosynthesis is water, not carbon dioxide. Photosynthesis are converted into two reactions: dark reaction and light reaction and dark reactions. NADPH is also formed during the light reactions, along with the ATP that is used for reaction chemical energy for dark reactions to form a particular amount of here is basic structure of chloroplast. We have to follow the basic structure of chloroplast, though it is not included in the syllabus, like a general structure of the leaves. Chloroplast are found in leaves, and leaves are the site of photosynthesis, and leaf structure is cuticle, epidermis, mesophyll, and vascular bundles. Then we came across the chloroplast, a cell of plant cells. It contains vacuole nucleic acid cell wall. And then we came across the chloroplast that contain outer membrane, inner membrane, double membrane structures, and thylakoid membranes that combine to form garena, and intermembrane space called stroma, and Thylakoid also contain interior space called lumens. These are the basic terms used for plastids or chloroplasts that are used in photosynthesis. It is enough for this chemiosmosis. This mechanism, photosynthetic pigments are the substances that absorb photosynthetic light, though it is not also included in syllabus that as carotenoids, chlorophylls, xanthophylls, yago, and carotenoids, red to orange chlorophylls, various type of chlorophyll A, B, C, and bacterial chlorophyll and bacteria. These are chlorophyll A and chlorophyll B differs only in their functional groups. This is chlorophyll A, chlorophyll B. Chlorophyll A contains CH3, like it contains CHO. It contains one extra oxygens and it contains three extra hydrogens. All other things are equals, and other is that chlorophyll is blue green and chlorophyll B is yellow greens. We have to understand particularly this type of reactions. Not all type of reactions is mechanism that carotenoids absorb light, they transfer it to the chlorophyll B, and chlorophyll B absorb light and transfer it to chlorophyll A, and chlorophyll A is the most abundant and directly take part in a chemical reactions. We have to understand this only. Carotenoid also protect our eyes. Most important point: light driving energy. Light absorbing the light is radiation of energy absorbed in the form of photons. Visible light is only observed at range in 380 to 750 nanometer in wavelength. Absorption spectrum is indicate the absorptions of the different pigments lights, like absorption of light from different pigment are shown by absorption spectrums, and action spectrum show the relative effect of these particular type of radiation that are absorbed. Action spectrums is obtained by illuminating plot with the light of different wavelengths and then estimating relative CO2 consumption, oxygen release during photosynthesis. Then we came across the difference between the action spectrum and the absorption spectrum. The peaks in absorption spectrum as compared to peak in the absorption spectrum, the peaks in action spectrum are broader, like peaks in action spectrum broader, and we come across valleys. These are narrow, peaks broader, and valleys narrow. Action spectrums and not deep. Valleys are not deep. Is action spectrum and absorption spectrum like this. It is absorption spectrum and it is action spectrum. And like absorption in the most observed range is more than absorption itself. Why absorption itself? Like at a particular wavelength, 600 or 500 nanometer, the absorption at green wavelength, the absorption is more than absorption itself, because many absorptions are taken by from carotenoids and chlorophyll B that increase the absorption spectrums, so the absorptions increases. It is most important point, the carotenoid and B also broaden the absorption spectrums of chlorophyll A. Then role of carbon dioxide, light independent reactions. We know that carbon dioxide is a basic reactant for the photosynthesis and is used in the light dependent reactions to form ATP and NADH. 10% of photosynthesis occur on the land plants and 90% on oceans. Air contains 0.03 to 0.04% carbon dioxide. That is a basic source for the land photosynthesis. And carbon dioxide enter into stroma and get dissolved in the mesophyll cells, and here is the route for the carbon dioxide to enter into the leaves. And these stomata are adjustable pore that are usually open during the day, and CO2 is required for photosynthesis. Stomata are open and carbon dioxide enter into the body for respiration. And for the plants that are in oceans, the source of carbon dioxide for them is the carbonates and bicarbonate dissolved in that, and daily rhythmic activity as stomata is due to the internal clock located in them. As we know about photosynthesis, reduction, oxidation reactions, and reduction reactions, like carbon dioxide is reduced to the glucose and water is oxidized to oxygen. But it is not a simple step process but composed of series of many chemical reactions, like light two basic categories: light dependent reactions which use light directly, and light independent reactions which do not use light directly. And light dependent reactions, energy is absorbed, and NADPH the reducing power and ATP the assimilating power are formed. And in the light independent reaction, these powers are used to form the sugar, the carbohydrates. Like light dependent reactions, this is photosystem 2 electron transport chain, photosystem 1 and light reaction, it form ATP and NADPH that is used for Krebs cycles, that is used in ribulose bisphosphate phosphoglycerate. And G3P is used for sucrose, and then it is cycle recycled, and NADPH and ATP is converted into the NADP and ADP. So and oxygen is here released in atmosphere. Sucrose is like sucrose and glucose, and carbohydrate are formed. Other amino acid and fatty acids also forms. Carbon dioxide is used in Calvin cycles. And light dependent reactions, energy is absorbed by photosystems, like pigments, and pigments are arranged like photosystems. Photosystem are basically divided into two systems: photosystem 1 and photosystem 2. Photosystem 1 is P700. It was discovered first, and it absorbed light of 700 wavelength. And P68 is the photosystem 2. It absorbs light of according to 680 nanometers. Photosystems contain a light gathering antenna complex and reaction centers. Antenna complex contain chlorophyll A, chlorophyll B, and C, and other carotenoids. Primary electron there, and in reaction center there is primary electron acceptors that accept electrons and transfer them to the reaction centers and follow them on the electron transport chains. There are two type of electron flows: non-cycle electron flow, in which ATP, ADP, and all oxygens are released, and cyclic electron flow, in which only ATP is used and photosystem 1 are used, and non-cyclic photosystem 1 and 2 both are used. Like we came across non-cyclic photophosphorylation, then this is the detail. Then light come at photosystem 1, and electron jump from its primary electron acceptors to the primary photosystem 1 to the primary acceptors. Like two electrons are shown, and these electrons come from hydrolysis of water, and water splitting step is called hydrolysis process of water splitting step process of this is called photolysis, and oxygen is released in it. Then at primary electron center, it is at higher energy, it came downward and decrease in energy, and this energy is used for formation of ATP. It came across primary electron circuitry to plastoquinone, then cytochrome complex. Here the its energy is used for ATP formation through chemiosmosis, then plastocyanins, a copper containing, and then it is entered into the photosystem 1, whose electron deficiency is created by the light, and whose electron is also in the primary acceptor of photosystem 1. Then it came across the pyridoxine iron containing, then NADP reductions form the NADPH. So NADPH oxygens and ATP are produced in this reaction. Sometimes the ATP get deficiency and NADPH and oxygens more. Then we are shifted from non-cyclic to cyclic, then photosystem 1 to primary electron to pyridoxine to the cytochrome complex again. And in this step the ATP are produced by chemiosmosis, and this process is repeated again and again until enough ATP is produced for the for dark reactions. So it is continuous. So only ATP is produced in cyclic process, and only photosystem 1 are used. Chemiosmosis is the process in which first of all electron is transported. You know that photosynthesis occur in the chloroplast. Photosynthesis occur in chloroplast, and it has stroma and thylakoid interior membrane lumens. First of all, the energy when the electrons came across down the this chains, electron transport chains, its energy is used to transport hydrogens from stroma into the lumen of thylakoid membrane, and hydrogens are stored in this stroma in this lumen. This is this is stroma, and this is lumen, thylakoid interior space lumen. And when cytochrome then electron come across cytochrome complex, then hydrogens are converted from stroma to lumens, and they are stored here in like H plus H plus H plus. And at the time of the chemiosmosis, when the ATP are produced by it, then we know that there are special channel called ATP synthesis. This is ATP synthesis enzyme or proteins that are in the thylakoid membranes. These and hydrogens came down the concentration gradient, and ADP is produced in these chemical reactions. So here is the mechanism where energy is produced by chemiosmosis. Then this energy, which is produced during the light reaction, is used in productions of specific sugars in a dark reactions. And this carbon dioxide is used, NADP is used, produced during the light reactions. ATP, 9 ATP are used during light reaction, and carbohydrate triose is formed. Only triose are formed. 3 carbon sugar are formed with this particular type of the reactants. If we have to produce glucose, which is of 6 carbon atoms, we have to double these, like 6 CO2, 12 amino ATPH, and 9 ATP are used. It is most important MCQs of MDCAT and other type of reactions. And this reactions was studied by Malvin Calvin, and it got Nobel Prize in 1961. And it is called Calvin cycle. At this particular type of scientist, particular name of scientist, it has 3 phases, like fixation phase, carbon fixation phase, reduction phase, and regeneration phase of ribose bisphosphate. First of all, we have a reactant ribose bisphosphate, 5 carbon 3 molecules that react with 3 molecules of carbon dioxides. And first of all, we came across the 3 molecule of 6 carbon compound that is unstable, and that unstable compound is broken down into the 6 molecule of 3 carbon compound phosphoglycerates. This is the first product. C3 pathway is called Calvin cycles because the first product that is formed is called phosphoglycerate, that is C3 carbon atoms. Phosphoglyceric acid first form, and then phosphoglycerate is formed like during the 1 bisphosphoglycerate glyceraldehyde 3 phos, and ribose bisphosphate reaction catalyzed by enzyme, and the enzyme that catalyzes this reaction is called Robisco. The product of reaction is highly unstable, a 6 carbon compound with no add 2 molecular carbon 3 phosphoglycerate. 3 phosphoglycerate is the first the product of Calvin cycles, so it is called C3 pathway also. Then it is the second phase is the reductions. It is the reduces. How it is reduces? By taking hydrogen from NADPH. First of all, there is the phosphorylations. 6 ATP are used here, 6 ATP are used here, and 3 phosphoglycerate is converted into 1 3-bis phosphoglycerate, and carbon phosphate is added to at carbon number 1, and there is phosphate at carbon number 3 already existed, like either your, and then second phosphate is added to carbon number 1, so it become 1 3-bis phosphoglycerate. Then it is reduced to glyceraldehyde 3 phosphate. 6 molecules of glyceraldehyde 3 phosphate molecules we have, and only one left the cycle to form various type of the sugars like glucose and other organic sugars, and 3 then 5 glyceraldehyde 3 phosphates are recycled in the cycle, and then ADP is used. Then they are converted into ribose phosphate. Here is the ribose phosphates. Then 3 ATP are used, one for 8 ribose phosphates to form ribose bisphosphate. The reactant of the original chemical reaction, and the reaction is recycled, and this phase is called regeneration of acceptor of CO2, because it accepts CO2 and form the particular type of phosphoglycerate.